Welcome to the Joanne Avison podcast and the second series of Sacred Geometry. We have spent 5 episodes now in the company of the dead. Not morbidly, gratefully. Da Vinci, Snelson, Fuller, Flemons. Each of them handed us a piece of the code and then kind of left the room, the way the great ones do, leaving the door open behind them. We're still left seeing how their respective genius lands in understanding the human body. And perhaps each one of us has provided a number in a combination lock that will somehow open the key to magic. I suspect it might, so stay with me. Something changes here in this episode because not only are we working with a living scientist, we're working with one who works with living bodies. This episode takes us into the world of the living and deep into rigorous science. Not the kind that dear Tom Flemons would call scientism. This isn't the opinion-based noise of outraged practitioners who want to give the impression they know what they're talking about because they cannot bear to think they've studied something, such as classical biomechanics, for decades that simply isn't accurate. Bless them. Nor is it sufficient that someone is famous. We are moving into the deep and genuine curiosity of a true scientist and researcher whose ability to honour the mystery of the human body inspired him to ask new questions. He isn't that well known, yet his research would break the spell of biomechanics if it was duly recognised. Not at the gross level of kind of outward expression in sports and exercise science necessarily, but more at the level of the cell, which renders everything else in a whole new light. For centuries, the body has been treated as if cells are units, like bricks in the walls of our living structure. One by one, they build us into wholeness by joining the dots of our separate parts. Tom Myers has become famous for taking those parts and suggesting they all join together to exist as myofascial continuities or meridians. Anatomy Trains is a great progression from bits to bands. The problem is simply that humans being are round. They're not made of bands or slings or train tracks. And while it represents progress of a sort, it doesn't honour the non-linear biologic forms we are as a whole terrain. It's progress with a small p, and it distracts us from roundness that we never fail to animate. We're all non-linear biologic forms, all of us. And somewhere in all this fame and friction is a deeper truth. Today, we're going to step out of history and into a laboratory that is still running experiments this morning, in a building that still has the lights on, with a man who is still right now, somewhere in Boston, thinking about the architecture of, let me say, any living cell. Donald Ingber is alive, a note that changes the frequency of everything this podcast will point to, because his work challenges everything suggesting linear organisation, even when it points towards continuity. It's a useful stepping stone that has to be permitted to grow or go in the face of whole body motion that we embody. Every one of us is round, and the principles governing the individual cell are true for the whole multicellular being. We are whole, not divisible into bits or bands. That's just what's true. Now, history has a kind of a stillness to it. We can turn it around in our hands like a coin worn smooth and safe to hold. We can storify it and we can challenge or accept a narrative, but no one answers back, at least not literally. But the living's not like that. The living is still moving. It's still arguing with itself. It's still capable of surprising us tomorrow, is still capable of changing its mind in the light of new discoveries. And Ingber is our first guide in this series who might, in theory, hear this episode. Let's make sure it's worthy of that possibility and honours the same huge respect for his curiosity that he has for every cell he's asked to guide him. Ingber's work, it's not hallmarked by an idea that he sought to impose on the architecture. It's governed by his willingness to ask the basic unit of life what it has to tell us and learn from it instead of imposing on it. That's different. And it makes an exquisite difference in what there is to learn from his research. Donald Ingber is the founding director of the Weiss Institute for Biologically Inspired Engineering at Harvard University. Also the Judah Folkman Professor of Vascular Biology at Harvard Medical School and Boston Children's Hospital. And he's also a Professor of Bioengineering at Harvard School of Engineering. He holds both a medical degree and a doctorate from Yale, and he has published hundreds of papers, filed hundreds of patents, founded companies, and been named among the world's leading translational researchers more than once. And none of that is why we are here. It's all the result of his curiosity, not the inspiration for it, or the road he took to prove that he was right. He asked better questions, and we're here because of an article that he wrote in January 1998 for Scientific American called The Architecture of Life. And I read that article and something in me recognised it, the way you kind of recognise a face in a crowd before you consciously know why, or the movement signature of a friend you haven't seen for years moving towards you at an airport. It kind of lifts your heart. You can see them before you can see them, if you know what I mean. It fills you with hope and it delights you at a level that just wants to shine in absolute joy. And Ingber's work had that effect on me. Now, this article, and get a copy of it if you can, wasn't written like biology or a master's degree. It's written like architecture or geometry, or like something Fuller or Nelson might have written. Except this time the domes and the cables were not made of aluminium tubing. They were made of us. Now, Ingber tells us the origin of this himself, and it isn't a story about him that someone else has reported. It's told in his own words about a time back in his undergraduate years at Yale in the mid-1970s. And that's where his studies of cell biology and his studies of sculpture were happening side by side. And it was there, he says, that he realised the question of how living things take shape has less to do with chemistry than with architecture, sculpture. Not art in general, but a specific discipline of shaping form with the hands. Think about that for a moment. The man who would go on to found an institute for biologically inspired engineering at Harvard, who would help create an entirely new scientific discipline, traces the beginning of it back to art and sculpture with his hands. Not his microscope, but to a room where he was looking at form and shape and structure for its own sake. Da Vinci, the artist and architect, Snelson, the sculptor, Fuller, the architect, Flemons, the fascinated craftsman, compelled to create models of what he knew was there, made by hand. And Ingber, looking at form, shape, and structure in an art class that engaged him in asking new questions. That's not a coincidence in this series. That is this series in one sentence. So what did he actually find? Before he ever ran the experiment that would make his name, Ingber built something. It was made up of 6 wooden dowels, some elastic string, arranged in a particular way. And he put the dowels in 3 pairs at right angles to each other, none of them touching, held apart and together only by the tension of the string. Exactly the same principle as Snelson's X-piece holds its own struts apart, yet it's complete. And then he sewed the ends of the dowels to a piece of cloth, stretched tight and pinned down flat to replicate how a cell might cling to a hard surface. When he released the pins, the whole structure sprang. Note this word specifically. It sprang upward into a rounded shape, puckering the cloth beneath it as it rose, precisely the way a living cell behaves the moment it's freed from something rigid it had been gripping. He kind of built the idea before he proved the so-called mechanism. Now, in the early 1990s, Ingber and his colleagues devised an experiment so simple it's almost comical to describe, and so significant that it helped to form the foundation of a new discipline. They went on from the art class experience to this simple experiment. They attached microscopic magnetic beads to specific receptor proteins sitting in the membrane of a living cell. Then they twisted those beads using a magnetic field from outside the cell entirely. The cell responded by stiffening the whole cell, not just the patch of membrane under the bead. The entire cell resisting the twist as one whole structure. They then isolated the founding principle of Tensegrity architecture, which is natural resistance to deformation. We are designed to adapt and sustain our shape. Now, the references for that, Wang, Butler and Ingber, 1993 science paper on mechanotransduction across the cell surface. That gives you the information to find it. It'll be in the notes as well. That single finding told Ingber something that classical cell biology had not considered or been built to explain. The cell was not just a bag of fluid with things floating in it responding only to chemical signals. The cell was a living structure, a living architecture that communicated via structure to and from within its environment. It's a tensioned, pre-stressed, self-stabilizing architecture in which a pull applied at one point was felt instantly everywhere. In one instant, using the kind of simplicity that Snelson encapsulated in his X-piece, Ingber discovered the keystone of Tensegrity architectures. They resist deformation in all directions. And just to confirm, levers can't do that, and neither can puppets. Your body is much more intelligent than any lever-based mechanism can ever describe. Just an inadequate explanation. Ingber immediately had a word ready for that particular kind of structure, because Fuller, as we've learned before, had already given the word to the world, and Snelson had already built its essential elements with his own creative hands. He cracked the code. Ingber activated it in a living cell. He knew he was witnessing Tensegrity in action everywhere. The big difference with Ingber's work was that it was not a sculpture in a gallery or even a dome over a stadium. It was possibly something that was happening inside every one of the roughly 70 trillion cells that make up you right now, while you listen to or watch this. Imagine that architectural intelligence working in you as we spend this time together. Who knew? And here's the point where Ingber and I would have a lively conversation about terminology. Unfortunately, for me, the word mechanics has become a synonym for motion in living bodies. For me, it's a violation of every strand of the archetypal poet in my being. I just long for the term biomotion to replace it. Not because I thought of it, but for the reasons I loathe mechanistic terms applied to the beautiful, unique beings I meet every day. We are not machines. We are living, messy, muddled, love, light and sound energy in motion, moving as our basic sign of life, with emotion, awareness and exquisite laws of nature, all bundled together with hope and happiness, seeking mostly to make the world a better place and wake up to what and who we really are. However, I love that Ingber wanted to at least rename his discoveries and change the term biomechanics. That's a win. The suggestion that this science applied to the living needed specific recognition. At least he has permanently changed the game, and that takes a lot. And much gratitude for the new questions he is asking. Ingber gave this new territory the name mechanobiology. It's essentially the study of how mechanical forces, not just chemistry and genetics, govern how living things develop, heal, and function. And he has said in his own words across decades that mechanical forces matter for biological control as much as chemicals and genes do. He said something very close to that in 1998. He is still saying it now in interviews given this year, 2026, not far off, 3 decades later. 30 years, and the sentence that distinguishes his inspiration and his conviction has not changed. That tells you something about a person. Some scientists change their minds every decade. Ingber found something true early. And true things don't need to change. And what this means for the body you live in is quite specific. Every time you stretch, every time a manual therapist works into a restriction, every time your foot meets the ground, you are not simply moving muscle over bone, the way the old lever diagrams suggest. You are transmitting a bioemotional, mechanobiological signal into the living architecture of your tissues. And to speak in Ingber's language, you're transmitting a mechanobiological communication through your architecture. That includes your individual cells, and those cells are, in a very real sense, listening, responding, and orchestrating your energy force through all of you. Every one of them is translating that pull or push or twist or that impulse into biochemistry, into gene expression, into the next choice or decision that cell makes about what to become, what to repair, how to work with its friends, the other cells of you, what to communicate through the in-between. The forces that you apply with your hands or your own movement are not separate from the chemistry of healing. You could say these force transmissions are one of the body's languages. I once heard it said that Tensegrity is the algorithm of the living body. I suspect that's accurate, but I don't honestly know exactly what an algorithm actually is. But I do know that if it likes something, it attracts it, and if it doesn't, it repels. That makes sense in a way. It bypasses the mentalizing and goes straight to the keys of bioemotional integrity. And I'm jumping ahead though, because I'm excited by Ingber's genius. This is where his work reaches directly towards everything this series has been building. Fuller drew the geometry in the sky. Snelson built it with cables and struts you could touch. Flemons brought it down into a spine you could hold in your hand. Ingber found it already alive, already working, inside every single cell, without waiting for anyone to design it there. It was there all along. Structure discovering itself at every scale, from the geodesic dome down to the ferromagnetic bead twisting a piece of a membrane of one of your own cells, through nothing but force transmission. Just think about that for a minute. What if we really have been under an actual spell for some huge number of centuries, and Ingber has unlocked the code applied to human beings in motion? What if mechanobiology can safely bypass biomechanics and actually make sense of living motion on every scale? Ingber's more recent work has carried this outward into organs on chips, tiny engineered models of human lungs, guts, and other tissues built on these same tensegrity principles, and they're now used to reduce reliance on animal testing and to study disease in ways that just were not previously possible. One of these designs now sits in the permanent collection of the Museum of Modern Art in New York. Back to art. Because essentially, we are creative creatures. Science, engineering, art, design. Ingber has said himself that his work has helped dissolve the boundaries between them. He would know. He walked through those boundaries himself. On the way from sculpture and cell biology at Yale to a Harvard laboratory that now studies the architecture of life itself. That article in Scientific American is gold. Ingber closed that same piece that he wrote in 1998 by reaching back through history rather than forward into the laboratory. He invoked Darcy Thompson, who had once invoked Galileo, who had once invoked Plato, arriving finally at a single thought Ingber chose to leave his readers with. That the book of nature may indeed be written in the characters of geometry. Which brings us at last to the reason he sits at position 6 in this particular storytelling sequence. Because everything Ingber found in the cell, John Sharkey has spent his working life finding, translating, and making sense of in the whole living human being. The same tension, the same continuity, the same reflexes, translated across the continuities of nonlinear biologic forms, the same refusal to be explained by levers and hinges and separate parts stacked on top of one another or strung together in lines. Where Ingber's laboratory is a single cell under a microscope, demonstrating these basic principles, Sharkey's laboratory, whichever one he's in, wherever he is in the world, has been the dissection table itself, the whole human form, opened with the same question in mind, with the same curiosity. Is this one structure communicating with itself in every direction all at once? It's a great question, and it's one that John Sharkey has asked through different modalities for the living and studied on cadaveric specimens for decades as a clinical anatomist and exercise physiologist. That's where we're going next. Only this time we're going to meet him in person, and he's going to be able to share that with us directly. Can't wait to see you there, and I cannot wait to talk to John. In the meantime, any details in this episode you will find in the show notes. Thank you for being here. And if you haven't heard the previous episodes in this series, or the series before it on the 5 Platonic volumes, that will give you a real sense of the elemental value of understanding the sacred geometry within all of us. Go back and see them in the Joanne Avison podcast on YouTube or on any podcast platform. It's really intriguing, and I pray it invites you to ask new questions. God bless.